Hello everyone, my name is Darlan Gusso. I am a PhD student from Pontifical Catholic University of Grande, Brazil. My advisor is Dr. Carla, Denise, Bonan. I'm here to present the abstract entitled Pyriproxyfen exposure impairs cognitive parameters and alters cortisol levels in zebrafish. Pyriproxyfen is one of the most used larvicides and insecticides. It acts as an analog of juvenile insect hormone like as a growth regulator. Therefore, it has been used as a strategic agent to combat the proliferation of mosquitoes, especially for mosquitoes of the genus Aedes. The pyriproxyfen is highly toxic during all stages of mosquito and can suppress metamorphosis and interferes in insect reproduction or proliferation. Despite the increasing number of toxicological studies about larvicides and insecticides, there are still few studies about the effects of pyriproxyfen in non-targeted species such as fish. Well, this study evaluated the effects of pyriproxyfen on behavioral, cognitive, and endocrine parameters using zebrafish as animal model. Initially, the effective pyriproxyfen concentration was assessed by mass spectrometry. We exposed adult zebrafish with 6 months to different pyriproxyfen concentration. The exposure regime was during 96 hours. And at the end of the exposure, the animals were submitted to analysis. Well, to behavioral parameters, we used novel tank test. To memory, we used inhibitory avoidance. To cortisol levels, we used ELISA assay. And to gene expression, for the glucocorticoid receptor GR and corticotrophin-releasing factor CRF, we used PCR. In this study, all protocols were approved by Institutional Animal Care Committee. Well, our results demonstrated that pyriproxyfen exposure didn't alter locomotor's parameter like distance or mean speed, neither unshit-like behavior that was evaluated by latency to pass to the top zone or time in the top zone of the tank. However, we could observe an impaired inhibitory avoidance memory at all tested pyriproxyfen concentrations. As we can see in the first graph, we haven't significant difference between training and tests in the exposed group. This could conclude that the fish have problem with memory. As you can see in the second graph, cortisol levels reduced in all exposed groups when compared to control or vehicle. So, we can see reduced cortisol levels, however, the GR and CRF gene expression following unaltered. Well, taken together, these findings indicate that pyriproxyfen may induce cognitive impairment and alter cortisol levels in zebrafish, that is a non-targeted species. To the best of our knowledge, this study is the first to report the effects of pyriproxyfen on memory parameters and cortisol levels in zebrafish. Moreover, in another study, Manuel et al. Showed that zebrafish with the highest level of cortisol performed better in memory tests 24 hours after training. Well, for our study, further research with this compound is needed to characterize the interaction sites between pyriproxyfen and endocrine and correlation with the central nervous system. Well, to finish, the present study underlines that zebrafish have the potential to be used in translational studies linked with learning and memory in animals that were submitted to chemical compounds exposure. I want to thank my advisor, Carla, Denise, Bonan, the Pontifical Catholic University of Rio Grande do Sul, and the financial support, CAPS, CNPq, and Impairs. Thanks everyone.